All right, so welcome everyone to the Longfin Smelt session. We have some excellent speakers today. And I just wanted, if you're just joining us now, I wanted to do a quick housekeeping. So you can join the conversation on Twitter with this hashtag 2022 IEP Workshop, like you can see at the bottom of the screen. And we're going to be kind of strict on time here to make sure we keep on schedule since we have concurrent sessions. So if you have questions during the presenters' talk, then please either type them in the Q&A box by clicking on the button at the bottom of the screen, or you can raise your hand at the conclusion of each talk. And as time allows, we will unmute the people that have their hand raised to ask their questions. So now we'll go straight into it. First up, we have Dr. Florian Mauduit, a postdoctoral researcher at UC Davis.
Good morning, everyone. My name is Florian Mauduit. I'm a project scientist working in non-fungi and Richard Connon laboratories, and today I will present some of our work on Longfin Smelt. More precisely, the study I'm going to present aimed to transfer approaches to evaluate the health of early life stages of Longfin Smelt. So since I am the first speaker of the Longfin Smelt session, I will start with a short introduction of the species. So the Longfin Smelt is a small fish in the family Osmeridae found along the Pacific coast of the United States from Alaska to California. In California, Longfin Smelt is historically found in the San Francisco estuary and the Sacramento-San Joaquin Delta, also called Bay Delta, the Humboldt Bay, and the estuaries of the Eel River and Klamath River. So the longfin uses a variety of habitat from nearshore water to estuaries and lower portions of freshwater streams. The longfin smelt are aerialine, meaning that they can tolerate a wide range of salinity from completely fresh to marine. They are also anadromous, depending on fresh and marine waters for spawning and rearing, and they typically live for 2 years, reaching a length of about 10 centimeters. The longfin also spawn occurs, the spawning events occurs from November through May with a peak from February through April, and they are usually considered as a semelparous, meaning that they usually die after spawning. I said usually because we found that some individuals can have multiple spawning events, but Levi Lewis might talk more about that later. However, as you can see on this figure representing the result of the fall midwater trawl survey, the population of longfin smelt in the Bay Delta has declined significantly since the 80s, and as a result, the species was listed as threatened under the California Endangered Species Act in 2009. The reasons for this decline are multiple and synergistic, and as listed in MOIL 2002 include a reduction in freshwater outflows, entrainment losses to water diversion, changes in food organisms, disease, competition with introduced species and predation, loss of genetic integrity, and finally exposure to toxic substances. However, now we need to understand how these threats contribute to the decline of longfin smelt population. For this, physiological tools and techniques have been allowing researchers since over a century to characterize how organisms respond to changes in their natural environment and how they interact with human activities or infrastructure. Over time, many of these techniques have become part of the conservation physiology toolbox, which is used to monitor, predict, conserve, and restore plant and animal population under threat. However, early life stages are often overlooked in conservation physiology, and most of these tools have been developed to be used on larger fish, so juveniles or adult organisms. So to fill this gap, we aimed to provide an approach and tools merging aspects of bioenergetic, cardiorespiratory physiology, and behavior to be able to assess the effect of stressors on the overall health of the larvae. To assess the effect of a stressor on bioenergetics, we can measure the size of the larvae and its yolk sac volume, which is a proxy for energy use over the first few days of life. So Yuzo Yanagitsuru, who will also give a talk later, published a paper on Longfin Smelt with this method. So briefly, to measure the yolk sac volume, we take pictures of the yolk sac over different angles, measure the dimensions with a calibrated slide, and calculate the volume using the formula for an ellipsoid. We also wanted this approach to include some assessment of cardiorespiratory physiology. So on larger fish, there is a test that I've used in the past that consists in measuring the heart rate of an anesthetized fish during a progressive temperature increase until fish shows arrhythmia. So you can see the setup we used on these pictures, and I'm sure you realize that we can do that on larvae. There is no way we can place electrodes. We can place the larvae on electrodes and measure the ECG. So Yuzo and I developed a setup to benefit from the fact that the Longfin Smelt larvae are translucent and that we can therefore record videos of the heart using a camera and a scope. So the video we record are then analyzed with a software that automatically counts the heartbeats, allowing us to measure the capacity of larvae to increase their heart rate with the temperature. This is an example of a video we recorded at 20 degrees, which is a high temperature for Longfin Smelt acclimated at 12 degrees. And I'm not sure that you can see the heart beating on zoom, but with the activity plot, you can see that the heart rate is irregular with periods of about 20 seconds without heartbeats. The last element we wanted to introduce in our approach was the behavior. For that, we used a setup developed for zebrafish larvae that allows to track the movement of larvae placed in microplates. A common high-throughput test commonly used in toxicology consists in verifying the response to a stimuli such as a change in light or tapping. But before that study, we didn't know if the Longfin Smelt larvae would respond to any stimuli. We know for a fact that some larvae don't respond to anything. For instance, we did some tests with inland silversides that hardly respond to light or tapping. To validate this approach, we decided to apply it to assess the effect of bifenthrin on Longfin Smelt larvae health. So bifenthrin is a pyrethroid insecticide used in agriculture and that we find in the river where it is extremely toxic for aquatic organisms. Bifenthrin, as all the pyrethroids, interact with voltage-gated sodium channels in neurons, which makes the behavior and cardiac function relevant endpoints to test its effects. So our experiment consisted in exposing embryos of Longfin Smelt from fertilization to hatch, which corresponds to a period of 18 days at 12 degrees. 4 concentrations plus a control were tested with 8 replicates per treatment and 25 individuals in each replicate. To give you an idea of the concentration we tested, last week we measured 10 nanograms per liter in sample collected at Rio Vista, and it can go even higher than that after a rain event. So to assess the effect of this exposure on larvae, we used the approach that I described earlier, combining aspects of bioenergetic, cardiorespiratory physiology, and behavior. All endpoints were measured at 24 hours post-hatch. Now I am going to present the results of that case study, starting with the bioenergetics. On this first figure, on the bottom left, you can see the size at hatch in millimeters of larvae from the different treatments. We observed a downward trend with the concentration increase, and larvae exposed to the higher concentration of bifenthrin were significantly smaller than those from the control. The second figure on the right shows the volume of the yolk sac of larvae from the different treatments. All treatment but the higher concentration displayed a reduced yolk sac volume. So this indicates that the exposure to bifenthrin had a significant energetic cost, and we can assume that this cost is probably detrimental for the survival of the larvae over their first few days of life in the environment. Also, not observing an effect with a higher dose is common in toxicology and can be explained by the fact that the organism is overwhelmed and its reaction inhibited. Now I'm going to present the result of the cardiorespiratory assessment. So on this figure, you can see that the heart rate increased gradually with the temperature, which was expected as fish are ectotherm. But this validates our method, that we are able to measure a reliable heart rate increase with the temperature. However, we didn't find any effect of the exposure to bifenthrin on larvae's capacity to increase the heart rate with temperature or any other endpoint measured, such as the temperature at which the heart rate peaked, the maximum cardiac frequency observed, or the temperature at which we observed arrhythmia. Finally, we tested the effect of the bifenthrin exposure on larvae's behavior by putting the larvae in microplates and tracking their movement over a 40-minute period with different periods in the dark and in the light. So on this figure, you can see the changes from the control for all the endpoints measured, such as the total distance moved, the swimming velocity, the cruising time, the freezing time, and the bursting time. So if the color is purple, this means that what we measure is higher than in the control, and if it's green, it means that the endpoint measured was lower than the control. So we observe that in the dark period and with the increasing concentration, we see that most of the endpoints were higher, meaning that the exposure to bifenthrin induced some hyperactivity. So to conclude, this study allowed us to verify that the endpoints measured were highly sensitive and informative, to the point that we have been able to measure the effect on an embryonic exposure. Remember that we exposed the organisms right after fertilization, and we measured the effect just after the hatch. So we saw a significant effects on bioenergetic and behavior, and although we didn't see any effect on the cardiac function, that endpoint was useful in other studies with different stressors. So finally, we now have some tools and an approach that are allowing us to better understand the requirement to the species to strive as well as impact of stressors. Thank you for your attention, and if there are any questions about my presentation, I'll be happy to answer them after this talk, or you can also email me.
Excellent. We have some time for questions now. If you want to type them in Q&A or raise your hand.
Hey, looks
Right, Ernest?
Yeah, can you hear me?
Yes.
Yeah, Florian, thank you for that talk. That was great. So lots of new information there. Just a question about I guess the changes when it comes to change in bioenergetics, but not in the cardiac in the cardiology of the species. Usually, what does that? Because I thought that was interesting. I mean, usually, what does it imply when you only have a bioenergetic change, but no change in the cardiac movement of cardiac rhythm of the fish?
Can you show the slide of bioenergetics? So basically, it was we measured the oil volume at the yolk sac volume at hatch, and that's like a proxy for energetics. So basically, this means that the exposure to the bifenthrin had an impact, like on their energy stock. So when they hatch, they had like lower energy reserves than the controls. But you're right, we didn't see any impact on the cardiac function. So I think it's because like these two endpoints are not really related. Like when we test the cardiac function, it's more to test the capacity of the organisms to transport oxygen to the organs. And so it doesn't really require, so the fish doesn't have the time to use like all these reserves. So that's not necessary, like both endpoints are not necessarily linked together. So that's why it can be like the exposure can affect one endpoint without affecting the other.
Sure. All right, thanks for explaining that.
A bit more time for any others. I see a hand, just working on whose it is.
Levi.
All right, go ahead, Levi.
Hey, Florian, great presentation. Quick question. I find your biomarkers really fascinating in terms of trying to understand and quantify, immediate effects on fish. I'm curious if you have any feeling in terms of the severity of the effects that you saw, how those might translate into kind of population level consequences. I know you can't directly do that, but if you were to say like these are super severe or not severe, what would you guess based on what you found?
So yes, that's hard, like to link that to a population level effect, because like for instance, we see a decrease in energy reserve. So I think this one is really like detrimental for their life, because this means that these larvae will need to be able to find food earlier than like their counterparts that have not been exposed. So that's like that's probably like detrimental for their survival, because these larvae are not necessarily able to catch food like 4 or 5 days post-hatch. So I think that's the main issue. The other thing is that like the hyperactivity, we try to, like we have studies to try to link that with a higher, like with ecological consequences, because we know that they are hyperactive, but what does this really mean? Does it mean that these larvae will take more risk to find food, for instance, and so will be more susceptible to predation? So that's one of the hypotheses. And this hyperactivity can also mean that these larvae needs more energy to live. So altogether, I think that it has like the capacity to make these fish more susceptible to predation, and at the same time, it also means that they need to find more food. So I think that it does, like I have the feeling that it has some ecological consequences, but this needs like to be demonstrated.
Yeah, thank you. That's fascinating.
And now I think it's time to move on to our next presenter. Thank you, Florian. We have up Dr. Levi Lewis, PI in the Otolith Geochemistry and Fish Ecology Lab at UC Davis.
Let's see. Are you able to see me, hear me?
Yes.
All right. Well, thank you. So I'm Levi Lewis. I'm the PI of the Odell Geochemistry Fish Ecology Lab at UC Davis. And today I'll be talking about some new insights into Longfin Smelt life histories. I'd like to first acknowledge my co-PIs on this project, Malta Vilmus and Jim Hobbs, as well as the many staff members and students that have been working on this project, as well as our many agency partners and funders, DWR, CDFW, and the Stewardship Council, as well as our many different collaborators at UC Davis, all of us are working together on addressing Longfin Smelt problems, just like Florian and I. So for this talk, I'd like to just share some of my perspectives on the Longfin Smelt life cycle, some of the key knowledge gaps, and how otolith techniques might be able to allow us to address some of those knowledge gaps. And then to share some recent kind of hot off the hard drive results based on otolith work that we've been doing in our lab over the last several years, specifically looking at growth, phenology, and migration. And then I'll spend a little bit of time arm waving about some of the management implications of our results. So Florian gave a great introduction to Longfin Smelt, so I won't spend a lot of time on this, but basically they live for, on average, 2 years, spawn in low salinity habitats, and they rear there for the first part of their lives, move out to higher salinity, all the way out to the ocean, at least some of them, and then completing that life cycle after 2 years. So they're in these coastal habitats, again, from here all the way to Alaska. And without going too much detail, two really interesting facets in the San Francisco estuary population have been perplexing researchers, conservationists, managers for quite some time. And that's the long-term decline in abundance, which, as Florian mentioned, could be due to many different competing stressors. And this really strong outflow abundance relationship that we've seen even as the fish continues to decline. And it's really clear based on this strong outflow relationship that Longfin Smelt need fresh water. They are a semi-anadromous, at least part of the population is semi-anadromous. And so if you take into account, this is just a really simple model looking at the long-term trend in the red dashed line on the left, and then modeling the variance around that trend as a function of the stock, so H2 fish, and then outflow. And basically, outflow drives the majority, or could it be used to explain the majority of the variance around that line. So freshwater outflow has a huge effect. But, we still don't really understand why. And we don't really know what life stages it's acting on, and we don't know the mechanisms. So Wim Kimmerer and many others have been working on this issue for a couple decades at least, and have identified many of the different hypotheses that could be driving these types of flow abundance relationships for Longfin Smelt and also for other species. And I've been working with John Rosenfeld to kind of distill this down into some really simple hypotheses that we can kind of adjust and describe and kind of update what the current science is thinking. And, outflow could affect ecological interactions like predation, competition, productivity. It can affect the quantity or extent of habitat, like volume or area of spawning and rearing habitats. Flow can affect, similarly, habitat quality, which is kind of linked to extent, but also the not just the amount, but how good the habitat is, based on a bunch of different types of parameters like temperature, turbidity, contaminants, food. And of course, flow has a physical effect on the retention, dispersal, and migration of fish as well. So these 4 general categories pretty much encapsulate most of the hypotheses about how flow is affecting Longfin Smelt. Well, what can otoliths tell us about some of these knowledge gaps? Well, otoliths are ear stones, and they're found in most bony fishes. To the fish, they have special functions that I talked about in my poem. To fisheries biologists, we like to use them because they allow us to study things about individual organisms. With a lot of massaging using jeweler's tools, we can get these otoliths into really nice thin preps. And then they provide us with daily increments that we can use to age the fish on a daily scale. The widths of the increments are often correlated with the growth rate of the fish, so we can get growth rates on a daily level. And we can also burn or ablate across the otolith from the core to the edge to get the entire chemical history of that fish and its environment. So it's a really powerful tool for describing the growth, phenology, and kind of movement history of individual fish. So just repeating what I said. So we collect these increment profiles and they allow us to look at age and growth. And from that, we can address kind of this habitat quality question by looking at how the condition of Longfin Smelt, for example, might vary spatially or temporally or as a function of various features using growth rates. And then we can also look at how reproductive phenology, such as the timing of hatching, might vary as a function of different environmental conditions. Similarly, we can also use chemistry to look at the salinity history. And using salinity history, we can infer movement patterns. We can use strontium in low salinity habitats to describe patterns in rearing and dispersal in the early life history. And then to look at later parts of the life history when fish are in higher salinities, we have to move on to a different tracer that has a broader salinity range of sensitivity, which is oxygen. I won't be talking about oxygen today. I'm going to focus just kind of on the juvenile and early life history for today. But I'm happy to talk about some of our other projects where we are using oxygen. And I always want to put a plug in that Leticia Cavoli, our postdoc, is putting in several proposals to continue doing the adult migration work on oxygen. So before we can use increment profiles, we need to validate that when we count increments, we're getting what we actually think we are. And I just wanted to give a shout out to Wilson Siu, who published a paper with us comparing known age fish from the FCCL, and then comparing the estimated ages from many different agers, and just showing that we get really high accuracy and precision when we use this aging tool. And we've also recently done an experiment where we manipulated salinity by trucking. Christian Denny talked about this in his speed talk, where we trucked seawater back and forth between FCCL and Bodega Bay and manipulated salinity to test our ability to reconstruct salinity history when we've experimentally manipulated it. And blindly trying to reconstruct salinity history, we were able to get the salinity values to within 0.5 part per thousand, and the timing to within about 6 days, which is pretty good for a fish that, when we're looking at juvenile fish, that could be 200 or 300 days old. So I hope I've convinced you that there are some important knowledge gaps for longfin, and otolith tools can be used to start addressing some of these. And so now I'd like to follow up by sharing some of the recent results that we've gained from our recently completed Prop 1, specifically looking at larval and juvenile longfin smelt. And so we looked at growth phenology and migration. So for the juvenile component of this, we archived about 500 juvenile longfin smelt that were provided by a variety of different sampling programs throughout the estuary. We had fish everywhere from lower South Bay all the way up to the North Delta. A lot of the fish came from the San Francisco Bay study, so big thank you to Kathy Heeb for archiving those for us. Obviously, this started with Jim Hobbs archiving fish over this kind of 20-year period. And many other partners have provided these. And we also went out and collected many of our own fish as well. We combined those with data collected and maintained by DWR on environmental conditions in the Delta, specifically temperature and outflow. And we used those data records to create interannual indices of winter temperature indices. And then also we used just the total outflow to create indices of outflow, which we use then to also kind of describe what some of the interannual patterns and growth might be. And then we did our magic polishing these otoliths, counting hundreds and hundreds of rings on every individual fish. And I just want to give a big shout out to Alec, Nico, Katie, and John Wang, all of our staff, and there are several others as well, who put a ton of time into this over the last 2 years. And so these are the results of their hard work. You can see size at age on the left. You can see that converted into growth rates. And we call these growth chronologies. So we actually have the timing of growth rates through time. And then we also have the salinity chronologies or the chemical chronologies on the right. So we put these into a general additive model and kind of took a first look at what some of the patterns might be if we look at variation in the growth rates of these fish as a function of several different factors. So these are those results, and I'll just briefly walk through them. So overall, growth rate varied ontogenetically, so with age from 0.1 to 0.5 millimeters per day, with kind of the post-larval pre-juvenile stage growing the fastest. When we looked at salinity down in the bottom left here, we found that fish in the when fish were in the lower salinity zone, so fresher or low salinity, they generally had higher growth than when they were in mid or high salinity. However, if you look at these 4 plots, there was an interaction between salinity and age. And to really simplify it, I've just pointed out these two arrows. On the top is the ontogenetic growth effect of fish that remained in freshwater. And in the bottom is the ontogenetic growth effect of fish that remained in high salinity. And so it seems that older fish tend to grow better at high salinity than low salinity. So this might be an age-salinity interaction that indicates some sort of either smolting or some other environmental correlate with salinity that causes growth to be poor in older fish later in the season or later in age. We also looked at patterns in geography, and we found that Sassoon Bay and South Bay generally had higher growth than other parts or other regions of the SFE. And this actually would make sense if wetland habitats are producing more food, as has been shown by Bruce Hammock in the upper estuary and Arthur Barras in the southern part of the estuary. We didn't see any clear effect of temperature. There's several different hypotheses I don't really have time to go into, and we can model it a couple different ways. But one thought is that maybe these fish are able to migrate, unlike delta smelt, to avoid the worst temperatures, for example, in the delta. And last, freshwater outflow, when we looked at growth rates between drought years and normal years, we saw higher growth rates in normal years, lower growth rates in drought years, and actually lower to intermediate growth rates in the most extreme flood years. And in many ways, this actually matches some of the predictions based on population flow models. So this could indicate that growth at least has some role in the flow abundance relationship. Okay, we also could look at phenology of all of these fish. So phenology is just the timing of events in an organism's lifetime. And we can quantify hatch dates simply by counting back from the edge, which represents the date a fish was collected and sacrificed, all the way back to the hatch check, and just simply subtract the number of days from the known collection date, and then you have hatch dates. We did this with all of our individuals, and these are the hatch date distributions from 1999 to 2017. And I failed to mention, we didn't have fish archived in all years. So this is not a continuous range of years. So there are some years missing here, clearly. And then we again combine that with DWR's water quality data. And we just ran a very simple model. First, we just looked at differences among years in terms of hatch dates. And so there was quite a bit of variation. Hatch dates range from -50 to 120 in day of year days, which is basically November to March, which matches exactly what we would expect. The median differences between years was up to 60 days. So the center of distribution of hatching can vary from year to year by at least 2 months. When we looked at outflow effects, we didn't really see clear differences in hatch dates as functions of outflow or even the interactive effects of outflow and temperature. There was some evidence in 2011 and 2017 that maybe outflow resulted in some later hatching and a broader distribution of hatching, but we just didn't have enough years to really tease that effect out of the data with any confidence. But we did see a fairly significant negative relationship between hatch date and the winter temperature index. And this is very similar to what we found for delta smelt as well. So basically, Longfin smelt are hatching about 14 days earlier per degree Celsius increase in the winter temperature index. And this may be nonlinear. You can see some patterns in the residuals. And 2013 was a weird outlier, and I can explain why, but I won't now. But overall, the pattern seems to hold, and it matches what we're seeing for other fish. And last, we also were looking at dispersal and migration, again, for the same group of fish. So the otoliths are just such a powerful tool because we can look at all these different metrics for every individual fish. So again, same group of fish, same profiles, but now we're combining the increment profiles with the chemistry profiles, and that allows us to generate this salinity chronology. And these are the salinity chronologies of our 500 juvenile longfin smelt in this study. And the first thing you can note is that this is a pretty complex life history. If these were salmon, you would see just this nice kind of sigmoidal pattern, and that's not at all what you see. And there's a couple of important things to point out. One is that you see fresh and brackish hatching, rearing, and retention. And when I say hatching, we may not be able to distinguish, whether a fish was born in fresh water and then within a few days ended up in brackish water. But there's very clearly a lot of fish that are spending almost no measurable time in fresh water. Also, we can see juvenile dispersal happening for almost all fish by between 100 and 150 days post-hatch. That matches the life history that we're seeing. However, we also do see some fish that have extended freshwater residence. There are multiple patterns that could be teased apart statistically using hierarchical clustering. We don't have a fixed number of how many patterns actually are. This is one possible way of teasing that apart. And if we assume that these 4 patterns capture a good amount of the variation, we can then look at how those might vary among different years and among years with different environmental conditions. So this was just one quick analysis where we compared the relative proportions of each of these different life histories. And just to mention the colors 1, 2, 3, and 4 across the top match the letters below F, G, H, and I. I just didn't change them. And the colors match. So the one interesting pattern I wanted to share is that it seems that in the in drought years, we actually see more of these extended freshwater life histories. And in the wettest years, we see more of these brackish. And in some ways, that's really counterintuitive because you think, oh, in wet years, you have more fresh water, you should have more freshwater spawn fish. But I've seen this pattern multiple times in the data, and I questioned it every time. And every time I see it, it's starting to build a story that maybe this is related to dispersal. So maybe we see more freshwater resident life histories in dry years because the fish are not getting physically transported out of the freshwater system and vice versa. Okay, so what does this all mean for management? We've shown, based on these otolith data, it suggests that fresh and low salinity habitats are optimal for larval growth. We've already demonstrated or discussed those habitats as being critical habitat for longfin smelt, and this is just another piece of evidence indicating that, yeah, low salinity habitats, good for young smelt. Habitat needs, however, change ontogenetically. So again, unlike, well, maybe similar to delta smelt, but maybe more extreme than delta smelt, it seems that freshwater habitats become inhospitable for longfin smelt as they age, and that is probably why they move. Sassoon Bay and South Bay seems to be supporting the fastest growth. And again, that might be an indicator of the benefits of wetland habitats, and that might encourage additional wetland habitat restoration for the production of food. And we didn't see much of an interannual temperature effect. I really want to explore that more before I make any kind of conclusions about that, but it could be, again, related to their ability to migrate out of the delta when it's inhospitable. And then last, drought was associated with lower growth. So reduced growth during drought conditions might be one of the mechanisms leading to the positive flow recruitment relationship. What about phenology? Well, reproductive phenology is very likely to shift earlier. We know this is true for like almost all species on the planet. I won't say all, a lot of species on the planet. We've shown it for delta smelt and the delta, and several other species it's likely to occur. The timing of management actions, therefore, probably need to take this into account. And so if we're establishing pumping restrictions or directed outflows, the timing of those are going to have to be tuned to environmental variation, both interannual and long-term. And then last, semi-anadromous may be an oversimplification for longfin smelt. And there may be some evidence of estuarine migrancy, and I think Yuzo will talk about that in his talk. And these complex life histories can yield resiliency. And accounting for this kind of life history pattern, it could be really important for management and understanding their population dynamics. And I'll happy to take any questions if we have time.
All right, we have one question in the chat from Arthur. He says, could the high growth rate in post-larval longfin smelt be due to changes in diet, being able to eat larger prey items? What size range is that post-larval classification?
Yeah, that's a great point, Arthur. I feel like you should answer the question since it was your paper. But we know that based on Arthur's work, we know that these fish around 25 millimeters are switching over to mysid prey. And that's pretty much right around where we see that acceleration in growth rate. So Arthur, I think that's a great question. And if maybe more of a great statement. Yeah, that they're prey switching at that time.
And I see that Wim's hand is raised. So go ahead, Wim.
Hey, Levi, great talk, as usual. A lot of stuff to swallow in a short period of time, but I need to pour over it. I'm wondering about the pattern. Well, we see this pattern, I'll talk about later, where the early larvae are just wafted out to sea, and sometimes they come, or not out to sea, but out to high salinity, and sometimes they come back. And I saw something like that, only it's the inverse in some of the fish that you were, that some of your profiles. I think the second or third of your patterns. the one in the middle. And I wondered if you have any idea about what, the low mid salinity one. A lot of them sort of dip down. You can, well, in panel H down below, you can see that, and to some degree in panel G, so that they're going to a lower salinity and then going to a higher salinity. And I wonder if that's because they're going out so fast that you're not even catching the first few days of growth, and they're going into higher salinity and then coming back to rear at lower salinity.
Yeah, I totally agree. So either there's several different competing hypotheses. Yuzo will comment on this. Two things. One, what you're saying could be absolutely true, is that they just move out of the low salinity or the freshwater habitats so quickly that we're not picking up with the chemistry. Two, though, Yuzo is going to show you, I think, that the larvae and the eggs actually do quite well in fairly high salinities. Yeah. So maybe there is estuarine spawning. We don't know yet, for sure. Yeah, but that's definitely plausible. And the retention, I mean, that big scatter that you can see on the left of this figure I'm showing right now with the low mid salinity hatching rearing, freshwater hatching rearing, I mean, that big scrambled mess, I think, really ties into what you've talked about quite a bit in terms of that all of that squiggly mess is retention, at least in my opinion. And I think it's really cool to be able to pick that out with the chemistry.
Well, I don't want to bore you, but I'll talk about it some more later.
Sounds good.
All right, we got to get to the next talk, but I see there are a few more hands. Sean, Mike, I think there's one more. So hold on to your questions and we could get back to them if we have extra time at the end. Next up, we have Bryan from the U.S. Fish and Wildlife Service.
Hi, my name is Bryan Matthias with the U.S. Fish and Wildlife Service in Lodi, and today I'm going to be talking about a project I have been working on with Vanessa Tobias, my co-author, on the assessment of environmental factors on apparent growth in Longfin Smelt. This project started as part of the ongoing Longfin Smelt species status assessment. And as it turns out that there's very little information available on Longfin Smelt growth patterns. And this is in spite of recent and ongoing efforts using otolith increment analyses to fill in this gap. Understanding variation in growth patterns is important because changes in growth patterns can have population level consequences. And this is because growth and more explicitly fish size is linked with both predation risk and fecundity. In general, smaller fish have higher predation rates because they fit into more predators' mouths. And bigger fish generally have higher fecundity. The objectives for this project were to quantify the strength, direction, and potential interactions of abiotic and biotic factors influencing the apparent growth of Longfin Smelt. The second objective was to assess for systematic or long-term changes in apparent growth patterns of Longfin Smelt. To address our objectives, we used a modified biphasic growth model that incorporated temporal variation using a combination of fixed and random effects. The data we used were Longfin Smelt length at age and a suite of environmental covariates including outflow, conductivity, temperature, prey availability, and density. Before I get too far into the model that we developed for this project, I wanted to talk a little bit about growth models in general. And if you've ever estimated growth patterns on a fish population, chances are you've used the von Bertalanffy growth curve. It is the most widely used growth model in fisheries, and it assumes a constant growth pattern over the entire life of the fish. It is biologically supported, however, criticized for its simplicity. If we take a look at the figure over on the right, this would be the top panel. On the vertical axis, we have length. On the horizontal, we have age. And as you can see with the von Bertalanffy model, it is just this nice smooth curve over the entire life. For this effort, we used the Lester et al. Growth model developed in 2004, and it is a biphasic growth model that breaks growth into phase 1 being linear growth and phase 2 following the von Bertalanffy. And as you see in the figure over on the right, the solid black line represents phase 1 linear growth, and the blue line represents the von Bertalanffy growth in phase 2. And that transition point between the two growth phases can be linked with major biological events happening in the lifetime of the fish, things like maturation or ontogenetic shifts. Next, I wanted to talk about temporal variation in growth. And Often when we develop growth models, we commonly assume growth patterns are constant across time. And for simplicity, let's call this the constant growth model. So if you go out and sample fish for 5 or 10 years, you get their length and age, and you develop a single model to describe the growth patterns of all of these fish that you've collected. You are implicitly assuming that growth remains constant across time. And if we kind of divide that out over time, looking at it as more of like a cohort effect, so taking a look at the figure on the bottom left, where we have length on the vertical axis, year on the horizontal axis, you can see that these growth patterns that we're assuming are just constant and static across time. We're copying the same growth model across each cohort and each time period. For this model, we included temporal variation in growth using a log normal regression on the monthly growth increment. And this accounts for variation in environmental conditions. And we can consider these in 2 different processes. So we consider the systematic changes, things like seasonal variation in temperature, or stochastic processes, such as prey population dynamics. If we take a look at the figure on the bottom right, we can see growth patterns that are a little bit more variable over time. We have, in this example, systematic seasonal changes over time, where we have poor conditions in blue and favorable conditions in gray, and then kind of those shoulder or in-between conditions in white. And you can see a little bit more of a wavy growth pattern from this type of model because we are accounting for these seasonal systematic changes. Next, I wanted to touch a little bit on the Longfin Smelt length at age data that we used. There were approximately 300,000 length observations spread across 5 different surveys between 1980 and 2020 that we used in this model. And for ages, we assigned age using a length frequency model that my co-author developed for a different part of the Longfin Smelt species status assessment. The maximum age we estimated was 48 months, and this represented cohorts from 1978 to 2020. And here in the figure on the bottom, we just have an example of some of the data that we had from roughly 2000 through 2010. And each of the different gears that we used are represented by a different color. And you can see with these patterns that there are clear demarcations between a lot of the age groups across a significant portion of their lifespan. The environmental data we used in this model came from various sources, including CDEC, Dayflow, the San Francisco Bay Survey otter trawl catch for abundance indices, and long-term zooplankton surveys for prey indices. On the right, we have 4 different figures representing temperature, outflow, abundance indices, and prey indices plotted across time. And this is just to show you what these data sets look like. And all of the variables we used in this model were logged and centered, but not standardized prior to use, with the exception of temperature. Temperature, we used data from a single point in the Kirkenes Strait, and we used a parabolic relationship, so temperature squared. And when we think about growth for fish, there's usually an optimal temperature that provides maximum growth. If we exceed that temperature, growth declines. If we get below that temperature, growth declines. And we actually estimated this vertex, or this optimal temperature for growth, within the model. And that's going to become important on the next slide when we talk about results. So there's a lot going on in this slide, and I need to walk you through it a little bit. So within this model, if you remember from before, we used a biphasic growth model. So phase 1 was linear, phase 2 followed the von Bertalanffy. So phase 1 was estimated between age 0 and 6, and phase 2 was age 6+. So we actually estimated this transition point within the model. So taking a look at the figure on the right, we have our covariates going down the vertical axis, so temperature squared on top, followed by log of outflow, followed by temperature outflow interaction, and so on as we move down. On the horizontal axis, we have a regression parameter estimates. On the left panel, it's phase 1 growth, right panel is phase 2 growth. The vertical dashed line is 0, so any of our points that have 95% confidence intervals that contain 0 are represented in gray, meaning that they're insignificant. Variables in blue are representing negative relationships, and variables in red, this pinkish color, are representing positive relationships. So if we take a look on top at temperature squared, we can see that for both phase 1 and phase 2 growth, we have negative relationships with temperature. So taking a step down to log outflow, we have a positive relationship with phase 1 growth and negative relationship with phase 2 growth. Taking another step down at temperature outflow interaction, we have positive interactions for both phase 1 and phase 2. And we can continue walking down this figure. But one of the things I wanted to point out is that the relationship between these covariates isn't always consistent across both phase 1 and phase 2, and often they're not. Often we see opposite effects of the covariate on phase 1 that we expect, or that compared to phase 2. Next, I wanted to talk a little bit about the temperature relationship, because this one gets a little confusing without actually seeing a plot. So on the bottom left, we have our growth rate in the vertical axis and temperature on the horizontal axis. And with this model, we have both phase 1 represented in the solid line, phase 2 with the dashed line, and each of these have the 95% confidence intervals in the shaded region. For phase 1, we see a positive relationship between growth and temperature. So as temperature increases, growth increases as well. For phase 2, we see this nice parabolic relationship where we have our highest growth at an intermediate temperature. And if we go to either extreme, we see lower growth than we see at the optimal temperature. Taking a look at some of the temporal trends across time, here we have a series of figures. On top, we have maximum growth rate. Next, we have length at age 0, followed by length at age 1 and length at age 2 as we move down. For maximum growth rate, this actually represents a random year effect. And we can think about this as additional variation we were not able to account for within the covariates. And taking a look at this figure, we see that there is a pretty decent amount of variation from year to year, but we don't see any significant trends over time. It's actually remaining relatively stable. Taking a step down to length at age 0, we see a similar pattern where we have a pretty decent amount of variation year to year, but it's remaining relatively constant. I should point out this little blip in the early 80s, and this one needs to be taken with a pretty big grain of salt. Sampling protocols for the surveys we had up until 1990 or up until 1995 did not include measuring small fish. I think the minimum length that was measured was either 25 or 45 millimeters. So taking a step down to age 1, length at age 1 and length at age 2, we see similar trends that we saw before, where we have pretty high variability from year to year, but consistent growth patterns over time. While I go over the conclusions for this presentation, I also wanted to show you some model fits because I always find them to be very interesting to look at. So here we have 4 different cohorts spaced about a decade apart. On the left side, we have the length at age plot. On the right, we have the residuals. So I'll let you look at those while I talk about conclusions. So the major things that we saw were that environmental conditions appear to be a major driver of Longfin Smelt growth patterns. We identified no systematic trends in length at age over time, and the growth rates that we estimated using this model were actually similar to those estimated using otolith-based methods as well. Lastly, I wanted to touch on some caveats with this model. The big caveat, or one of the big caveats, is the Longfin Smelt migration. Within the data set that we used, there were no observations from outside of the Bay Delta. And one of the big things that we're going to need to do in the future is looking at the model bias with this. Are the trends that we see an accurate representation of what's actually happening out there? So one way we can do this is with otolith increment analysis, and the other would be to expand our spatial sampling so we actually collect fish from outside of the Bay Delta region, so out in the ocean. And the second one is that we did not explicitly account for changes in growth during early life. So we didn't account for transitions between the post-hatched, post-yolk sac to early juvenile transition. This model might do a decent job representing those transitions, but we need to do some more work to actually look to see if those are. With that, I would like to thank some of the people that helped with this project, including Joe Miller, who was a fellow over the summer with the U.S. Fish and Wildlife Service that helped compile a lot of the environmental data, and the Longfin Smelt Species Status Assessment Core team. And with that, I will happily take any questions.
All right, 5 minutes for questions. I see Levi up first.
Great. Here. Great talk, really fascinating. I want to dive into it some more. Maybe I can follow up with you after this. The question that I had was, I guess, statement question. One is, one of the benefits of using Odylus is that we can kind of tease apart time, size, age relationships. I'm just curious if you've thought much or accounted for, the potential variation in hatch dates. So for example, if in 1 year, most of your fish hatched in, November, December, and another year, most of your fish hatched in March, April, what is that going to do to your, size at age 0, size at age 1 estimate?
Yeah, so I've thought about this one a lot, and that's actually kind of captured at least partially within the length at age 0 estimate. We're estimating that using a series of covariates. I think it's I keep changing, or I do a bunch of model comparison, so I can't remember exactly what variables. I know there's a variable related to outflow during the spawning season and temperature during the spawning season, and an interaction as well. So the way that I've been interpreting that parameter, for the model itself, we're assuming a constant hatch date. But if the length at age 0 is higher or lower, we can interpret that as hatching either earlier or later in the season. So we're not explicitly accounting for differences in hatch dates, but the model is accounting for some of that variation.
Yeah, that makes sense. Just quick follow-up. If, I think there's two interpretations, right? One is, the size at age 1, for example, could be interpreted as differences in growth rate or differences in hatch date, right? And so I was curious how if you were trying to tease it apart, or if you were giving more leverage to one versus the other.
So those parameters are estimated jointly within the model, so there's no real easy or convenient way of actually separating those two out.
Yeah, that makes sense. Okay, thanks.
All right, R. Baxter. You're now unmuted.
Hey, Bryan. Thanks, cool. I'm glad you finally got to that. I played with the data a long time ago, but didn't get nearly as much out of it. I had a question, and I'm having conductivity problems, but did you look at abundance and growth rate?
Yes, that was one of the covariates that I used. It was I think the otter trawl, San Francisco Bay Otter Trawl Index, and we also had that interacting with month of year to try to account for some of the density dependent effects over time. And both of those were significant, but some of the relationships, actually looking at the estimates, were a little bit wonky. Sometimes we had a positive relationship and others we had a negative relationship. And I don't remember off the top of my head which direction they each went. But we are accounting for some density dependence, or attempting to.
Yeah, and a quick follow-up. In my look at the data, 1982, which was one of those big decline points, I think, and 83 were high abundance years and low size at age years, dramatically low. So those tended to drive the data set for what I looked at way back when, and they may be outliers on your analysis too. You may want to look in that. Anyway, thank you. Cool stuff. Glad you're able to do it.
Thanks. I'll check out those earliers too.
Okay, thank you, Bryan. Now we'll move on to the next one, which is a presentation from Dr. Wim Kimmerer from the Estuary and Ocean Science Center at San Francisco State University. And when we can see your slide, but we can't hear you.
Okay, can you see my screen now?
Yes.
All right, I'm not getting the presenter mode. Let me just get that up. Okay. Right. So abundance of organisms in estuaries vary with flow a lot, lots of estuaries, and a variety of mechanisms are possible to explain those. And I think longfin smelt may have the strongest relationship of any pelagic organism to flow. So I want to talk about that, but first a little background. You've probably seen these diagrams before. At low, medium, and high flow, you see the movement of the salt field as represented by the colors. And the main point here is that it's very dynamic. When the salt field moves seaward under high flows, you get stratification because of the interaction between the bathymetry and the flow. And also, this is all very strongly tidal, and the tidal dynamics have a huge impact on this. So, so when pelagic organisms encounter different salinities in different regions, they'll move differently. The bottom waters under high flow will move mostly landward, and the bottom waters under low flow are mostly under tidal control. So X2, you probably already know, distance up the axis of the estuary, and that's a really good indicator of the response of the estuary to flow. And it's geographically oriented, so it's handy. So here's the problem, or the challenge. We have this declining abundance. The red dots are the data, and the model is in blue. And the statistical model is combines the X2 effect and the year effect. And the X2 effect has a dynamic range of about 100-fold. So that's a lot of variation to explain, and we're going to try to explain it. So the life history, we need to sort of tag everything to life history, and the earlier speakers have been talking about that. So we start, this is essentially the life history here. We start with early larvae developing into later larvae, and then juveniles, and then the second go around, they develop into adults, and then they spawn in the winter. So, or winter to early spring. So we want to look at that in terms of the different sampling programs. We've got all these sampling programs that happen at different times. The smelt larva survey in blue, the 20mm survey, the summer townet survey, the fall midwater trawl survey, and the San Francisco Bay study, which has two different nets, the midwater trawl and the outer trawl. And these latter three have larger, well, the mesh sizes go up as you go around because they're targeting different stages. And so the sampling gear is the largest, has the largest mesh in these three studies here. That makes a difference because these earlier surveys don't catch everything. So this is the 20mm survey in a dry year and a wet year, and they miss the higher salinities, plotted against salinity, catch against salinity. And the same with the smelt larva survey. Under the higher flows, they miss the higher salinities because of the way they sample. So that makes a big difference that'll show up here and later. This is the timing. So what we did was we took the X2 slope from that earlier graph for the fall midwater trawl index and applied it to each of the different surveys. And the red line represents the slope you would get from the index. And the dots are the different surveys by month. The colors don't make any difference, but the surveys are represented by the shapes. And the main point here is that around May, the different surveys start showing the slope that's the same as the slope in the fall midwater trawl index. The earlier surveys don't show it. And to some extent, it's because of their sampling bias. And when you take out that sampling bias, I think you would see that the slopes start to gravitate toward this minus 0.1 or so. So what happened in May? What goes on in life history in May? And I'll take a look at some different mechanisms because you got to have the right magnitude and the right timing and the right place in the system to make the proposed mechanisms work. The size of the habitat. So the blue ones are going to be habitat effects. The size of the habitat does not support the mechanism because its magnitude can't be high enough. The same with this early dispersion of larvae. And the same with structural complexity. I'm not going to talk in detail about these. Food availability lacks the persistence because it doesn't cover the entire time series from 1967 to present. Spawning habitat doesn't match the timing, and ocean conditions don't match the timing. So then looking at physical dynamics, export entrainment doesn't work because we point this out in 2 papers that have been submitted, and it doesn't support the magnitude. It's a very small effect. Juvenile retention is too late in the life history to explain the May arrival of this flow effect. And then finally, larval survival. Well, I assert yes, and I'll show you why in the next slide. So here's that low flow pattern again, and the life cycle involving larvae moving rather rapidly down estuary and then sinking into the water column and then coming back up. And I'll show you some particle tracking work later that supports this. At high flow, they start further down estuary and they move much faster. And at some point they have to sink into the lower water part of the water column to be able to move back up estuary. And that movement's very rapid too. So I'll show you some data that support all this. Okay, so this is from Lenny Grimaldo's work. In a dry year, 2016, the larvae are found in Sassoon Bay primarily. And these vertical lines show a salinity of 2, essentially where X2 would come to. And then in 2017, a wet year, they're down in San Pablo Bay and they're close to, or actually a little bit seaward of the low salinity zone. So, so they go with the salinity field, and these later larvae, and we study them in their frame of reference by following salinity. So then, where do they spawn? I'm sorry, I missed something there. Yeah, I forgot about the local spawning in San Pablo Bay, inferred from the distribution of sizes. Okay, so now we have the smelt larva survey from January to March, 20 millimeter survey from March to July. And this is salinity by length, box plots, about 24,000 fish here and 200,000 fish here. And what you see is that the salinity distribution increases with age or with size until about 8 millimeters, and then they gradually decrease, implying that in respect to the low salinity zone, the fish are moving rapidly into the low salinity zone and beyond it, and then moving back into it. So they go with flow, and then they stop. But we're missing the higher salinity data in these graphs, so we don't really quite know what's going on here. So it's time to turn to particle tracking. This is from a hatching analysis that Ed Gross did. This paper has been reviewed, and we're working on a revision. And these are passive particles released in the region of the estuary that had the highest, we found had the highest hatching rate. So this is for 2013 flow. And these particles were released over 2 weeks. And then this is when they were recovered at the end of that 2-week period. So they're moving down the estuary, and they're moving very rapidly. Some of them are actually going out into the ocean. So why does that matter? Well, this is how far they have to get to. This is how far they go, I mean, in 3 days travel time. So why that arrow? That's the center of the smelt larva survey catch during that time. So that means that they're really moving rapidly, and a lot of those larvae are moving well past the region where they end up being seen later on. So how do they get back there? So we have to go and look at releases of particles down in the Golden Gate, and that's this pattern. So if you release particles at the Golden Gate, we released 62,000 particles. Well, Ed did, and after 1 month, they were mostly out in the ocean. Hardly any of them went back up here. So if they're passive particles and they're released at the Golden Gate, they go out to sea. If they're passive particles and they release up in the Delta, most of them go to sea. So what do you have to do to get them back up into the upper estuary? Well, give them behavior. These are sinking particles. All they do is sink at 1 millimeter per second, 2 millimeters per second. So they move back up into the estuary, and they end up mostly around here at 1 month. And that's kind of where we see them. That's where we see the larvae. So how does that work? Or what does that do if you change the flow? So this is for a flow of 730 cubic meters per second. I'll show you the time pattern of the particles that were released under different flow conditions. And these are synthetic steady flows. So this is the 730 line. So this is how long it took. It took 30 days, and most of them were back up in that region of the estuary where salinity is less than 6. So this is the proportion of the particles in salinity less than 6 over time. Particles released at the Golden Gate. And the highest flow, 1,440 cubic meters per second, they got to less than salinity in about 12 to 14 days. And at a salinity or at a flow of 100 cubic meters per second, it took them quite a long time to get up there. And by the end of this model run, at 60 days, they were not there yet. So why does speed matter to survival? How come it makes a difference if they eventually get there? The answer is right here. This is predation risk. Sorry, I've got a bit of a cold here. Predation risk based on the abundance of anchovy, which are the biomass dominant of small planktivorous fish in the lower estuary. They're very rare at low salinity, but they're much more abundant at high salinity, at medium to high salinity. And there are two different estimates here. One is based on some experiments done on an anchovy in South Africa, getting clearance rate, which is the amount of water cleared of prey per unit time. And then extrapolating it to the population using the base study catch data. And you can see that on average, about 40% per day, which is an appallingly high mortality rate to a small plankton-rich fish or a small larval fish. The red is from a different set of data, although I used the base study for the anchovy abundance, but it's anchovy bioenergetics and abundance and zooplankton abundance. And so they cleared about 35% per day. So it's very similar result. There are a lot of caveats here. For example, the base study, the nets we know don't catch all of the fish. They catch about half the number that we see on sonar. And there are a number of other things. They may not feed by picking at night. They may feed by filtering. So there's a number of caveats, but I'm going to skip over those for now, because this is all a work in progress. So what causes longfin smelt abundance to vary 100-fold with freshwater flow? I think it's our conceptual model. And as I said, it's a work in progress. We have a lot of things to do with, I hope, for future funds, like figuring out the triggers for downward swimming, more details about how they've returned from oceanic salinities. And you know, especially when they swim down, at what life stage. Expand the suite of behaviors. Look at the vertical distribution of larvae, nighttime distributions, effects of food, and predation estimates. So all these things come into play in this. And that's what I have. And I thank you for paying attention. And thank you for funding, and to all these people here. And I'm done.
Thank you. I saw a hand go up and then down.
Should I stop sharing?
Yeah. Oh, I see. Mike Eakin. Go ahead.
Hi, can you hear me?
No?
Yes.
Yes.
Okay. Thanks, Wim. Great talk. I had a similar question for Levi that I think is also maybe relative to ask you here. What do you think the role of egg transport plays in this relationship you're describing? You know, in theory, we don't talk about it a lot, but obviously eggs don't weigh as much as anvils. They're going to get transported by flow too. And I'm curious, you know, what assumptions and how we handle that sort of going forward.
Well, Levi was just schooling me on that, so I'll let him answer, because I don't really know.
Okay, great.
Well, I mean, we don't have the answer for the estuary here, but we do know there's a couple of papers written about longfin smelt spawning in Lake Washington, and they point out that the I don't know, for those of you that aren't familiar, longfin smelt have eggs with adhesive stalks. They can spawn those onto, you know, big solid structures where they don't move, but oftentimes they'll spawn in like very kind of like coarse sands, and so it basically results in the egg attached to a small weight. And it almost makes the egg neutrally buoyant, slightly negative. And they show that they basically bounce along the river where they're spawning until they hit a deep pool and they aggregate. So that kind of comes back to your question, Mike, if that's happening on a large scale in the San Francisco estuary, I think what that would suggest is that fish may be spawning higher upstream, eggs may be moving down to the area to areas downstream before fish actually leave the egg. I don't know if that really changes a whole lot in terms of what Wim is showing us. But I think that's actually been discussed. So we just don't know if it's actually happening.
If it happens, they would aggregate it in the low salinity zone. And if they hatch there, they'd already be home.
Right. And Yuzo, I think Yuzo's, I'll keep throwing you out there, Yuzo, but I think Yuzo's going to talk about egg and early embryo tolerances of salinity. And I think it suggests that the eggs can actually tolerate some fairly high salinities.
Next up, April Hennessy. You're now unmuted.
Oh, thank you. Can you guys hear me? Okay. Thanks, Wim. Great talk. Unfortunately, I lost internet a couple times during it. I think it's because my kids are home and streaming. But anyway, I just wanted to look at the timing that you had for the different life stages. And, you know, this year we did start smelt larval survey earlier. And we basically started detecting larvae in mid-December. So that was pretty exciting. And then here it's almost April and we're still detecting 6 to 8 millimeter larvae this week. And then even up to 30 millimeter juveniles. So it seems like there's quite a bit of overlap and quite a bit of diversity in that spawning window and the hatching timing and stuff like that. So I read your abstract and it seemed like you were saying that outflow in May is most important and X2 position in May. And so one of the things I was wondering is if we don't have as much outflow in March and April to hold that X2 position, I'm curious, won't that actually take more water to move X2 further down in May? And I apologize if I missed this because my internet went out.
I didn't say anything about the management implications, so I should have. I think, since the X2 standard was based on a bunch of fish, but really Longfin Smelt is the one we are most concerned about, I think it's time for some adaptive management experiments with outflow. And yes, I think taking all the outflow you're going to get for the season and dumping it down the rivers in May, and out of the Delta in May, might be a very interesting thing. But yes, the drier it is, the further X2 is landward, the more flow it takes to move it a certain amount. So, and it can get very, very expensive in water if it's dry.
Yeah. Okay, thanks.
Thank you. All right, last in our session, we have Yuzo Yanagitsuru, and he's a PhD candidate from the Fish Conservation Physiology Lab at UC Davis.
So I gave a very similar talk last year at IEP. So this is essentially an update on some of the work that we've done in order to improve conservation aquaculture, and I'll be expanding this a little bit to include some work that could inform conservation management strategies and actions for longfin smelt. So just to recap a little bit of previous longfin smelt physiology research, a lot of this was primarily aimed towards improving the conservation aquaculture program at the Fish Conservation Culture Laboratory, or the FCCL. So here we have the survival and percentage of longfin smelt in captivity to 3 different ages, each of these 3 different colored bars, for every year that longfin smelt have been brought in to try to culture them in captivity. And shaded in the blue is are the years where we improved the protocol used for rearing these fish based on some of the experiments that I shared last year. And what's apparent from this is that the survival in 2019 to all 3 ages was higher compared to the previous years, and most importantly, you can actually see the bar for 300 days post-hatch. So there were, at least relative to the previous years, quite a few fish that survived to what is essentially adulthood. Now, 2020 was a poor year for brood fish capturing. That was a COVID year, right? So probably not too surprising there. But in 2021, we seem to be following a pretty similar trend where we have survival to 40 days post-hatch, pretty much the same as in 2019. So this makes us pretty cautiously optimistic that this culturing change that we made based on experiments is actually working. And I'd like to just recap some of those, just the major results of those studies that I presented last year, which were focused on just 2 parameters. This is temperature and salinity. And we'll start with temperature here on the left. The major goal of this was to assess how temperature affected the hatching performance, growth performance, as well as survival of longfin smelt larvae. And there's a lot of arrows here, but the main thing to look at here is that in the 15-degree group, there was a lower hatch performance, there was a lower growth rate, there was a lower yolk resorption rate, so they were not able to utilize those endogenous resources that they're born with, and there was higher mortality earlier on. So based on this, 15 degrees is a bad temperature to rear these early life stages, the embryos and the yolk sac larvae. And based on these results, we had a very simple conclusion, which is that temperatures of 12 degrees or less are probably appropriate for culture. And this is indeed what is used at the FCCL today. The next was salinity. So here on the right, and this one has even more arrows, so I'll just guide you through this. We threw these fish into these 5 salinities, and then we measured water balance, salt balance, growth rates, yolk resorption rates, as well as mortality. And a few things that I'll highlight here. First is that there was decreased mortality or improved survival in these 5 and 10 parts per thousand salinities. Secondly, at 20 and 32 parts per thousand, which are of the higher salinities, you have a decreased water balance, or decreased water content, meaning that they are not able to maintain water balance properly, and salt content increase. So they were also unable to maintain that salt balance within themselves. So they were being physiologically challenged at these higher salinities. This translated to a lower growth at these salinities. So clearly these larvae are unable to tolerate these higher salinities. But interestingly enough, even though the freshwater environment, the larvae had they were able to maintain that water balance and salt balance, they also had lower growth performance, and this was translated, or this could be explained by a lower yolk resorption rate. The reason for why these fish are unable to maintain proper yolk resorption rates at freshwater is unclear, but there does seem to be some negative effect of freshwater on these fish. So based on all of this, we concluded that these moderately brackish salinities, about 5 and 10 parts per thousand, were appropriate for culture. And these 2 studies together were what really informed these culture protocol changes that seem to be very effective at improving survival. So now I'd like to continue on to what we've done since then. The first thing that I'd like to go talk about is an additional study on how we improved conservation aquaculture, or at least provided some insight for improving conservation aquaculture by looking at how turbidity influences the culturing of the larvae. So turbidity being just about the murkiness of the water. And for this study, it was a very simple design. We took these larvae and put them into 3 different turbidity groups of 10, 20, and 40 NTU. These are turbidities that are on the lower end and about on the lower end of the San Francisco estuary. So we did that, and we were feeding them at FCCL using typical feeds, rotifers, as you can see on the left, and Artemia on the right. And we measured survival, growth, and feeding incidence over time for these fish. Starting first with the less exciting results. Here is survival in percentage over the 3 time points that we measured these fish. And there was no difference in survival at these 3 turbidities. However, there was a trend towards higher survival in the 40 NTU, or the higher turbidity group. Now, what was significant, though, was the growth of these fish. So here on the left, we have the total length of the fish, again, at the 3 time points that we sampled at for the 3 turbidities. And you can see that by 10 days post-hatch, the 10 NTU fish were smaller compared to these higher turbidity groups, and that persisted at 20 days post-hatch. Now, what's interesting, although it wasn't statistically significant, even at 4 days post-hatch, that earliest time point, there was a reduced growth, or at least a reduced length, or at least trending towards reduced length for this lower turbidity group. And what seems to be the explanation for this reduced length is just lower feeding performance. So here on the right, we have feeding incidence in percentage. So the percent of larvae that were actually successfully feeding at the 3 different ages. And only on 40 DPH do we see an actual difference where the 10 NTU group had a lower feeding performance. So what we think is happening here is that the feeding performance even prior to 4 days post-hatch, we're not actually entirely sure when these fish begin feeding, but we're hypothesizing that these fish are actually feeding better at these early stages at these higher turbidities. What appears to be the case, though, is that once these fish are a little bit older, not that much older, they feed just fine, no matter what the turbidity is. So what the implication for this is in the culture, you could theoretically decrease the turbidity after a short period of time, which has the benefit of, one, decreasing the cost of production in these at FCCL, and also reducing biofouling in the tanks, because the turbidity, how we induce it, is by putting in dead microalgae, which can cause a lot of fouling in the tanks over time. Another major result here was what these fish seem to prefer to eat as they grow. So here on the left, we have the number of rotifers consumed for every feeding larvae over the length of the larvae. And these solid lines indicate the smooth regression of how many rotifers were being eaten as the fish grew. And these shaded areas indicate at what point the amount of rotifers being eaten changed. So what that means is that there was a change where they began to, in this case, eat fewer rotifers. And what seems to be the case is that for the lower turbidities, you have about 8 millimeters in length is when these larvae begin to feed less so on rotifers. This is mimicked over here on the right for Artemia, which is a larger feed, where about 8 millimeters in length, they start transitioning to feeding more heavily on Artemia. And so this informs some feeding protocols that could be altered at the FCCL, where fish about 8 millimeters in length, perhaps that's when we should be putting in some Artemia, because they are capable of feeding on these larger prey items. So to recap these turbidity results, the major results are, first, survival does not seem to be influenced by turbidity, but using higher turbidities prior to 4 days post-hatch could improve growth in culture. You could also maybe lower the turbidity afterwards, and you may not have any negative effects of that. Another major result is that the transition to feeding primarily in Artemia begins at about approximately 8 millimeters in length, which corresponds to about 5 to 7 days post-hatch. So transitioning these fish to Artemia could happen fairly early. Now, obviously, we know nothing about the digestive physiology of these fish, so whether or not this is actually good to do is still something that should be looked at. Now I'd like to transition to a study that we conducted to provide some insight for management strategies. So this is another study looking at temperature. So the design here is going to be very similar to what I presume Florian will have talked about. I think I'm the last talk here, which is to incubate these embryos at 3 different temperatures, hatch them out, and then put them into this procedure where we perform a cardiac assay on these fish. So what we do with these assay is we take these larvae and we expose them to a stepwise increase in temperature, and we measure the heart rate of these fish at each of these steps, these stepwise increases in temperature. So you can see the heart beating on this fish in this video here. And you can do this for a lot of fish at the same time. You can simultaneously measure the heart rate for all these fish using a video analysis software. And so this is a really high throughput assay for measuring essentially the cardiac performance of the fish at different temperatures. There are a few different metrics that are very useful that come out of this cardiac assay, and I'm just going to focus on two of them. I think Florian will probably have mentioned the Arrhenius breakpoint temperature, but I'm just going to talk about the indicators of the thermal limit, which are first T peak, so the temperature at which the heart rate reaches its maximum, and this is considered a sublethal thermal limit. Okay. The second one is TR, which is temperature at which the fish become arrhythmic. So you can see this in the video here on the right. The heart is beating and then it stops. It's currently stopped and then it'll continue beating again. It's a very arrhythmic heartbeat, and this is considered a lethal limit. Now, when you collect all this data and you create a box and whisker plot, this is what it's going to look like. So on the left, we have T peak, so that's sublethal limit. And on the right, we have TR, the lethal limit for these fish. And one of the major results here, which is completely unsurprising, is that at higher temperature acclimations or higher incubation temperatures, as I'd called it previously, you get higher T peaks and higher TRs. This is just one of the cornerstones of thermal physiology. This is not surprising. But one really important aspect of the results here that you cannot ignore is that the thermal limits are extremely variable across individuals. So you can see the whiskers are just enormous on these box and whisker plots. What that means is that if you use a median T peak or TR, one of these thermal limits, and say this is the temperature at which fish can no longer handle, and you were to use this as a guideline out in the field, you would really be having a lot of the fish suffering as a result. So instead, we decided to look at this through a proportional analysis. So when you plot the proportion of individuals at these limits over temperatures, you get a curve that looks like this. It's an S-shaped sigmoidal curve. And with this, you can get a few thresholds based on perhaps what you consider feasible for managing in the estuary. So here on the left, we have T peak, so that sublethal limit, sublethal thermal limit. And on the right, we have the lethal thermal limit. Each of these horizontal dotted lines correspond to a certain threshold percentage where you have 10%, 50%, and 95% of individuals experiencing thermal stress, whether that's sublethal or lethal. What that means is that you can create these guidelines based on these percentages. So 10% is obviously the most cautious temperature to use. 50% indicates a temperature where any increase will lead to a very rapid increase in the proportion of individuals that are at their thermal limit, because the slope of this increase in the proportion is the highest at this 50% range. And then the 95% threshold indicates where the you would have a catastrophic event, essentially. These virtually all the larvae are going to be experiencing some sort of thermal stress. So based on this, I won't comment on what exactly the temperature that one should use is, but I will say that one really big mystery right now is in the field, what temperature is a longfin smelt embryo exposed to? And I think that's a question that we don't really know the answer to. But it has this really big implication, though, because if you were to say use a threshold of assuming that these embryos are reared at 12 degrees Celsius, you would 12 degrees Celsius, and there was these embryos were actually incubated at 9 degrees out in the estuary, you're really going to increase the proportion of individuals that are going to be experiencing thermal stress, whether that's the sublethal or lethal limit that you decide to choose. So under the precautionary principle, it's probably safest, given the current information, to use these lower incubation temperatures and the thermal thresholds that they dictate. So just in summary, we used these turbidity experiments, and we just discovered that about increased turbidity, at least at the earliest life stages, could improve longfin smelt conservation aquaculture. And using this cardiac assay, we were able to describe a set of thermal thresholds that could be used to guide management strategies. Yeah, I'd like to thank everyone who helped. So lab managers and technicians, Dennis, Heather, Mackenzie, Wilson, obviously all the FCCL staff, Luke, Galen, Troy, Zach, Elliot, Tori, Jill, Charles, as well as Delta, and then finally the Hobbs lab and other broodstock collection teams. There were several of them that were able to make this project possible, as well as funding sources, CDFW, U.S. Fisheries Wildlife, and the Delta Stewardship Council.
We're at time, but we'll get in one quick question for Yuzo from in the Q&A chat. We have Gonzalo, and they ask, any interaction effects of temperature, salinity, and turbidity on longfin smelt growth and survival?
Yeah, so that's a really important question to answer. We have not looked at that yet. That would be a really good next step, though. One of the issues with working with longfin is that to do a interactive effect study, you need way more fish, double the fish. So we haven't quite gotten there yet. But as we kind of improve the collections out in the field and are able to better keep these fish alive, we can answer those questions.
And I see a question from Michelle in the chat. We are over time, but it is lunch now, so maybe we can quickly get to it. Michelle,
Yeah.
maybe you want to, maybe I'll unmute you and you can summarize this. Where'd you go? All right, Michelle, you're unmuted.
Yeah, thank you. Yeah, sorry to go over lunchtime. I'll try and make it quick. It should be pretty easy to answer, I guess. From recent studies, we're starting to really see, and maybe we knew this before, but the recent studies really seem to show that they seem to be quite selective on copepods specifically, even when things like rotifers are abundant in the environment. Has FCCL tried to culture copepods yet and run experiments feeding them other things than the lab rats like rotifers and Artemia that they may not actually like that much or thrive on that much? I won't say like, I'll say thrive. Yeah.
Yeah, so I love that question because it's one that I'm pretty obsessed with. I think that we really should try culturing copepods. The one problem with copepods right now is even in commercial aquaculture, it's extremely difficult to just culture this food item in high enough densities to actually make it feasible for a high intensive rearing protocol. So to answer, the short answer is no, we have not tried raising copepods to feed these fish, but I do think that's a really, really good and really important next step. In terms of the mysids, so they have been culturing mysids. That's also a very difficult one to culture because they cannibalize each other, so you can't raise them at high enough densities to actually feed to the fish. But they tried feeding those mysids to the adult broodfish, and for whatever reason, they don't take them. It's very bizarre. Instead, they seem to prefer that lab rat prey of brine shrimp, Artemia, which is, you know, why? No idea, but they do seem to not eat mysids in captivity, at least the adults. Yeah, so
That's
really good question.
Cool. Thank you.
Okay, one last thank you to all of our speakers again. This was great. And now there's a lunch break, and this Zoom link here resumes at 1 o'clock with Delta Salmon.